Lastly, folks, I want to show us a graph of the kinetics of PFK. In this chart, we have three lines that I want us to look at. In this first line here, there are no inhibitors for PFK, so we imagine the concentration of ATP is low. In this next line, we have ATP and AMP. And in the last line, we only have ATP. So in this last line, folks, you see that it is very S-shaped. Where have we seen S-shaped curves before? When we were talking about cooperativity. So here we have another example again of where an inhibitor is increasing cooperativity. This is very interesting. Why does ATP increase cooperativity? Well, let's take a structural look and understand the structure function happening here. I want to remind us, folks, that the interaction between one active site and one allosteric site is happening between two different monomers. Let's look back at this slide real quick. Whenever I was drawing the active site and allosteric site in my cartoons, understand that the active site was in one monomer and the allosteric site I was drawing was from a different monomer, okay? So let's go back and draw that in real quick and then get to the slide that we were on. So the active site's from one monomer, allosteric site is from a different subunit. We'll also draw that in here for the ATP. But why does that affect cooperativity? Well, let's kind of zoom into the structure here to understand what's happening. So, in this picture, folks, the central helix is here. So, here is our central helix that I was drawing in on my slides. And I'm going to erase that just so that the slide is a bit more clear. Okay. Now, what do I want us to look at? Well, I want us to, excuse me, I thought I turned my pen on. I need to fix this in my slide, huh? This is glue. So I'll fix that for you now. In the allosteric site, we have ATP bound. In the active site, we also have ATP bound, but no fructose 6-phosphate. You'll see this glutamate is sticking into the active site. That's why we can't have fructose 6-phosphate in there. It would repel through Coulomb's law. But we can also use Coulomb's law to understand what's going on in the allosteric site. Arginine is positively charged. ATP is negatively charged. This would have a strong, favorable, attractive association. But the association is happening between two different subunits. So what's on the left is from subunit 1, and what's down here is coming from subunit number 2. So this attractive association through arginine to ATP, the attractive association of one subunit with another subunit is what is increasing the cooperativity. Okay? This increase of cooperativity is what we're seeing because of this attractive ionic interaction through Coulomb's law, with a positively charged arginine in one subunit interacts with a negatively charged ATP in the allosteric site of another subunit. And so that's why we see this increase in S shape when we add ATP to the curve. Now, of course, you can also see that when we add AMP, the curve shifts back left, so we can understand that AMP is actually an activator. A few final very small notes here, folks, because we're seeing this enzyme modulated by AMP, ATP, ADP, right? Our body keeps really tight control over ATP concentrations, but flux through glycolysis can vary by 10,000%, right? It's kind of wild. ATP concentrations do not change by any more than 10% at any given time. Our body's really good at maintaining homeostasis for levels of ATP because we always need a lot of energy, right? So how can flux through glycolysis change so much? Well, what happens is that the concentrations of AMP and ADP are actually changing a lot more dramatically than concentrations of ATP. So for example, ATP is always between 0.9 and 1 millimolar, while ADP is between 0.1 and 0.2 millimolar, which is a doubling. And AMP is between 0.02 and 0.1, oh no units, millimolar, which is a factor of 5. So we have much more ability to control flux by bigger changes in ADP and AMP, which is pretty cool. Finally, folks, I'm going to mention a couple of things to us to keep in mind. Of course, there is substrate cycling in glycolysis. Remember, most rate-determining steps we think of as being irreversible because they have such a large negative delta G. But we still need to be able to reverse the metabolic pathway. Well, we do this by introducing a new enzyme at the point of the rate-determining step, and we call it a substrate cycle. For example, we can reverse the reaction performed by PFK with an enzyme called FBPase, fructose 1,6-bisphosphatase. You might think of this as wasteful because that allows us to just go back and forth in the pathway, but actually this makes the cell adaptable, able to maintain homeostasis really well. As the last aside point here, folks, we'll be following glucose from start to finish through metabolism, but keep in mind that we eat many other types of sugars and we can metabolize those through glycolysis as well. That wraps it up for today, folks. What we did is we learned a little bit about anaerobic fermentation. I want us to keep that in mind because we'll use that knowledge when we discuss flux through these pathways. But we really focused in on PFK. How is it regulated? How is flux through glycolysis modulated through PFK? And we learned some structure function details of how that happens. I also reminded us that substrate cycling is happening in glycolysis. We just haven't seen it. and leave you with your cadmium. See you next time.